The overall goal of these psPACT and mPACT methods is the fast clearing of whole CNS tissue without the need for an electrophoretic tissue clearing system.
This method can help answer key questions in the human disease research model field, such as Alzheimer's disease, organ injury, and regeneration and tumorigenesis. The main advantage of this technique is that the mouse CNS tissue is not damaged by the electrophoretic tissue clearing and is rapidly cleared by modified passive clearing.
We first had the idea for this method after using the original PACT and past method for brain clearing. Visual demonstration of this method is critical, as psPACT and mPACT methods are difficult to run, because this method has multiple stages of sample preparation.
Begin by preparing PACT cocktail solution by adding VA044 powder to a solution of 4% PFA and 4% acrylamide at a final concentration of 0.25%. After perfusing the tissues with PACT solution, completely immerse the whole brain and spinal cord into a 50 milliliter tube containing chilled PACT solution and store at 4 degrees Celsius for 24 hours. 24 hours later, transfer the tissue to a 50 milliliter tube containing fresh cold PACT solution and connect the tube cap to a tissue gel hybridization system connected to a nitrogen tank and set it to 37 degrees Celsius. Then, turn on the vacuum.